Hi, my name is Olle Hernell, and together with my colleagues Susanne Lindqvist and Lennart Lundberg, I am the founder of the startup company Lipum, aiming at developing a new biologic pharmaceutical directed towards the protein bile salt stimulated lipase. The idea is to treat chronic inflammatory diseases with this new drug that has a completely novel mode of action. My interest in this protein, the bile salt stimulated lipase, actually goes back a long time when I, together with my supervisor, discovered the bile salt stimulated lipase in human milk. And we could quickly show that the enzyme had all the characteristics needed to be active in hydrolyzing the milk lipids in the intestine of the breastfed infant. We could also show that the enzyme is secreted also from the pancreas, or rather that it is identical to an enzyme already known to be secreted from the exocrine pancreas, at that time called cholesterol esterase. Sometime later, we also discovered that BSSL is present in the blood. And initially, we thought that it was absorbed from the intestine into the blood, but then we started to question whether it could have another source, actually, and started to look for that. And to make a long story short, we found that it was actually present in white blood cells, in granulocytes. And we could also see that when these granulocytes were activated, they secreted BSSL. And that evoked the question, could actually BSSL also have a different function to have something to do with inflammation? And to quickly check that, we used a very well-established model in mice. The collagen-induced arthritis model, and we had access to mice that were knocked out. The BSSL gene was knocked out, so they were devoid of BSSL, and we could see that compared to wild-type mice having BSSL, the knockouts were protected from developing arthritis. And that, of course, evoked the next question, could actually an antibody inhibiting the effect of BSSL, could that have the same effect to reduce the inflammatory process and the inflammation? So we checked that with the antibodies that we had available and found that yes, that was the case. And we started a very fruitful collaboration with Sci-Life Lab in Stockholm and Uppsala to develop a humanized antibody towards BSSL that could be used to treat inflammation in humans. And that has been a very productive collaboration. And actually, just a few days ago, we received the first batch of our candidate drug called SOL-116. And now we have started the first experiments to confirm the efficacy, again, in the same animal models. And then we will move on to do safety and efficacy studies in other species to finally, hopefully in a couple of years, to reach the first studies in the human. And we are very optimistic that that will work. Thank you very much.